cellular molecular level, and we have some wonderful people here doing research at that scale. Myself, I'm sort of a systems neuroscientist. I'm interested in how networks of brain areas work together to carry about, let's say, vision or hearing, or in my own case, motor control, how we move. Now, we also have a large community of neuroscientists here in the sort of cognitive and behavioral neuroscience domain. This may be investigating questions about how the brain changes across an entire lifespan. You can use all sorts of neuroimaging techniques to look at degenerative disorders like Alzheimer's and Parkinson's, or to look even at how the brain changes in the earliest stages of life. So neuroscience is really big here at Western. We have lots of great people. We have lots of great equipment. There's a really vibrant computational aspect as well that complements each of these scales of study. So just some facts about the program. We are fairly large, around 190 students, half of whom are master's students, half of whom are PhD students. We're across 5 or 4 different faculties. So the medical school, or Schulich, that's where we are right now. Large component of researchers in the Department of Psychology, which is the Faculty of Social Sciences. We also have Faculty of Science, so computer science and biology, and then even Faculty of Health Sciences, kinesiology, physical therapy, and things like that. So pretty much anything you're interested in, there will be research being done here on campus. We also have over about 100 supervising faculty as well. Now, for many of you, you may be considering why grad school as opposed to some other options. We are motivated in the program that as much as we want to teach you about our area of study and the things that we love, we're also in the business of developing scientists. So it's not just about going on into academia and eventually replacing us. It's about teaching you the skills that you need to succeed in society, regardless of where you go after you leave here. So I'll talk about some of the skills that you're going to be gaining that are going to translate well to wherever you go. Now, why neuroscience here, and why neuroscience writ large, and then why here at Western? You can't go a day, it seems, if you're paying attention to the media to hear something that actually impacts or relates to brain function. Whether it's, let's say, the brain fog after long COVID, what goes on in your brain when you're constantly distracted by devices and things like that, the plague of neurodegenerative diseases in society right now. Neuroscience is everywhere. We realize so much about what we don't know about brain function, so there's lots of things to study. Now, here at Western, it's a signature research area, and that means there's been lots of hiring of faculty and lots of purchasing of some terrific equipment here that many of the collaborators use, so for example, to image the brain non-invasively. We'll be hearing a bit about that today. And so this is an exciting field. It is highly interdisciplinary. We need all sorts of different skills to be able to study brain function at this time. So speaking of those skills, I think one of the best things I talk about when I'm sort of thinking about grad students and grad studies, your thesis is your project. You become a project manager, and this is just one of those skills that you're going to need that will serve you well regardless of where you go. Because guess what? You got to manage projects. You got to manage people, your time, your resources, and things like that. That is a skill. It takes time to develop it. We're helping you, or we're going to help you support. We're going to help support you to do that. So you'd be supported, of course, in your lab environment, your supervisor, any lab mates that you would have in that environment as well. But there's all sorts of other ways that the program supports your development too. For example, through advisory committees you meet with regularly to discuss your progress. And then there's all sorts of, whether it's seminars, whether it's courses, whether it's other aspects of the grad experience, you're learning all sorts of things. How to manage people, how to communicate science, how to explain complex ideas to different audiences. It's a different way of talking about things if you're talking to people that are familiar with neuroscience or if talking with your family at home. The way you bring them into your story is unique, it's different. So these are skills that you're going to learn if you come here. Ultimately, critical thinking skills as well. So we do have some courses that you take. There's a 9500 is sort of a critical thinking course. There's a seminar course, and then you also have the option to take some courses that pertain really to your direct area of study. So as you're contemplating and thinking about coming into the grad program here, or really anywhere in grad school, one of the probably biggest questions is, how do you find a supervisor? And this is somewhere where you probably have to do a little bit of self-reflection just yourself. What sort of neuroscience are you interested in? I talked about all the different scales here, but what really do you find the most fascinating? What sort of methods are you interested in? But then it also asks you to think a little bit about what sort of environment do you want? There are some labs here that are quite large, and correspondingly, you may or may not spend as much time with the supervisor. You may spend a lot of time with other lab members. In contrast, other labs are quite small, and it's a much more one-on-one environment. There's no one fit that's right for everybody. Rather, the right environment is out there for you. It helps if you sort of think about what sort of environment you think you would thrive in. So there's nothing wrong with starting to approach supervisors now. We're just starting our recruitment. The recruitment opens on November 1st, as I'll talk about in just a second. But another thing you can do while you're kind of going through the self-reflection is to go visit this website here. Remember I said there's like 100 supervisors? Well, in this site, we sort of grouped by research theme. So there would be a group on systems neuroscience, how you hear or see or whatever, a group on motor control, there's a group on molecular and cellular, and on and on and on. So I think there's 9 different themes that we've sort of clustered our supervisors in to help you kind of hone in on the things you find interesting. You'll find links to websites, etc. So as I mentioned, applications are coming soon. They open actually November 1st. And I'm going to go through a bunch of questions in just a second, but what you need to apply to ideally academic references, send in your transcript and your CV as well as a statement of interest of what you find interesting about neuroscience, a little bit about your background, so you're telling us where you're coming from. Average approximately 80%, I'll talk about that in just a second. One thing I do want to mention for the domestic students who would be coming in at the master's level, there's actually an opportunity to already apply for a prestigious scholarship. The CGSM is the Canada Graduate Scholarship. It's through NSERC or CIHR, if you've heard about that. But you actually will have an opportunity to apply for that. And so it's sort of encouraging you, if you are interested in applying, come talk to us in the neuroscience program about how you would also apply for that if you wish. Okay, so I'm almost done talking about the program itself, but let's just go through some frequently asked questions. We're going to stick around after as well, so I'm happy to talk about these as well. So one question we always get, do I have to have a supervisor to apply? It's actually a 2-stage process. We first screen you for eligibility, and then to come into the program, you have to find a lab that's going to take you in. But that being said, nothing is preventing you from going to talk to supervisors, potential supervisors right now. And in fact, if you already have that relationship, you can mention it in your application. Say, hey, I've already talked to Dr. So-and-so and he or she is willing to take me on. That allows us to know that a little bit of homework has been done and that it's a potentially good match. Okay, so nothing preventing you from doing that right now. Do you really need 2 academic references? In a perfect world, yes, because they can speak to your suitability for graduate school. But some of you, particularly perhaps because of the pandemic, might not have that sort of academic reference. So in those sorts of scenarios, you can have another reference that would speak to your character, speak to the things that, relate to success in graduate school, dependability, punctuality, what you did, projects you managed, and things like that. We're very flexible in the program when we're assessing an application. So yeah, that's so ideally academic, but it's not mandatory. Let's put it that way. So my background is not in science. Not in neuroscience, or let's say biological sciences or psychology. Can you still apply? That is an emphatic absolutely. We actually take in a lot of students from an analytical background, whether it's someone like an electrical engineer. Guess what? The brain's an electric organ. Those are really useful skills to have. We also take lots of people with a computer science or mathematics background in order to deal with the large data sets that these new technologies are providing. So with that, we actually have a sort of special route for people without a neurobiology or psychology background. Just for the first term that they're in the program, they take sort of like a small course to get them up to speed quickly, taking actually a second-year neuroscience course that allows them to speak and understand the language of science. We find that these are sometimes our best students because they learn what they need to know really, really quickly and then apply it with their other skills. One more list of frequently asked questions. You do not need an official transcript at this stage, but if you're accepted, you will have to provide them. So unofficial is fine at the application stage. What do I do if I'm a little bit below 80%? And this is where context matters. It's not like a hard cutoff. 80.1 is in, 79.9 is out. But if you do have some lower marks, explain that in your application. And there may be other things in your application that speak to your suitability for graduate school. So address it directly. You know, I didn't like this course. I didn't get a great mark in it, but hey, look at my marks in other courses. And not only that, I have this research experience that turned out great. Well, that'll allow us to look at we really do assess the application as a whole. Last question, what is required for the application? Well, we talked about this a little bit already, but the website here is a really good place to start if you want further information. So I'm going to switch gears now. Again, this is, I think it's just the same website, basically. I'm now going to talk about my own research and just spend 5 minutes on the projects that I'm looking to recruit for. So when I start this sort of talk, I think about an inverted pyramid. I consider myself to be a scientist, and in fact, I'm a neuroscientist. I wouldn't be in the Neuroscience Grad Program if not. There's lots of flavors of neuroscientists out there. Myself, I'm a sort of circuits neuroscientist. I'm interested in how networks of brain areas work together to carry about a function. The function I'm interested in is motor control. I'm interested in how the brain controls movement. But there's lots of different movements that the brain makes through the muscles in the body. I'm actually specifically interested in orienting. I got into this business studying saccadic eye movements. And those of you who are neuroscience students in Neuro 2000, remember that I was kind of a geek and I made you look at my eyes really, really close because I love that type of stuff. But lately, I've been getting more and more interested in not only eye movements, but also how those movements are coordinated for orienting carried about by the whole body. Now, in my lab, I have complementary research streams going in humans and in monkeys, non-human primates. I study how we can sort of mess around a little bit with this system to understand how it may change in disease. I'm working to develop some non-invasive modes of brain stimulation. And I'm also getting really interested in reacting when time is of the essence. How does this system produce movement when you only have a fraction of a second? And there's lots, we do this all the time in our day when you're walking and maybe something's going to affect your path, or if your cell phone's sliding off your desk, you reach out and grab it before you even think about it. Sports gives all sorts of great analogies for this as well, or examples, I should say. I'm a baseball fan too. This is the dreaded comebacker to the mound. And obviously in response to a looming visual stimulus, there was a really potent movement that happened that in this case probably saved the guys, if not, well, serious injury at the very least. Slow motion, or I love this picture because you see how quickly, this is a whole body response. This is your arm, your leg, your neck, everything's coordinated within that fraction of a second. The reason I find this interesting is because, both of these pathways, how vision gets into the brain and how we send motor control signals to the muscles, are really, really complicated. On the left side, these are some of the brain areas that put together the brain's analysis of the visual world. On the right side are all the brain areas that are communicating with the spinal cord. But there does appear to be a shortcut, this area in our midbrain called the superior colliculus. This is what I've spent much of my scientific life studying. It gets vision actually straight from the retina and also projects really potently, not only to eye movement centers, but head movement, arm movement, and things like that. So rather than firing baseballs at people in the lab, we have them conduct these sorts of video games. This is actually my dad. I recruited him. His task is to catch the ball that emerges if that big square is green, and if that big square is red, to try to move away in the opposite direction. Why do we find this interesting? Well, we also measure muscle activity. That's the wires coming off here that allows us to tell really precisely when the brain sends the signal to the muscle. And he was recruited for a study actually in Parkinson's disease patients. And we're finding that the first wave of recruitment, which we think is coming through not the motor cortex, but the superior colliculus. In Parkinson's disease, this disease where it's sort of like an absence or a paucity of movement, that first recruitment is actually higher than the subsequent recruitment that comes later, which we would presume is coming from the motor cortex. So it speaks to kind of a selective pathology in the disorder that selects one pathway or that impacts one pathway, but not another pathway. So that's basically it. You can find more information in my lab website. This came from a moment when we were actually throwing axes, and my lab, I guess they let the boss win, and that's why I'm sitting on the throne. But happy to talk more after. And with that, I think I will turn it over to Haley and Ashley. Thanks. Yep.
Hi everyone, my name is Haley. I'm the president of Songs and I'm here with Ash. And we're just going to tell you a little bit about our organization. So we were founded in 2017. We're a group of trainees spread across the neuroscience program on campus that's aiming to unite all of the trainees who are located at different buildings and different disciplines. And so we may not always get to see each other and create that sense of community. And so Songs currently consists of about 50 members in an executive committee, as well as subcommittees that organize events for the neuroscience program to create social and academic community. And now I'm just going to toss it over to Ash to talk about the different subcommittees we have.
There we go. Okay. Hi, everyone. As Haley mentioned, my name is Ash and I'm the vice president of Songs. So I'm just going to tell you guys a little bit about the different committees that we have at Songs. So the first committee I'll talk about is the Science Communication Committee. So the main objective of this committee is to run a neurograd journal called the Dorsal Column. So often students will write articles for a lay audience so they get practice with their scientific writing. We also have the Science Outreach Committee. So the goal of this committee is to just increase access for neuroscience information to younger students. So in the past, we've gone to high schools and given like mini lectures to those students. We also have the Big Ideas Committee. So this committee has been evolving over the years, but it's a good opportunity for students to come out to, view some presentations, panel discussions on research either being conducted here at Western or just some hot topics in science. We also have the Presentation Workshop Committee. So this committee helps students get feedback and get practice on their seminar presentations. So in your first year, you don't actually have to present at seminar, but it will become helpful in your upper years. And we're also trying this year to broaden the workshop so that people can practice for conferences and potentially also for that Neuro 9500 course when you have a presentation to give as well. We also have the Information Sessions Committee. So this committee is really great because they hold different information sessions on academic related topics. It's also broadening this year. So we're hoping to have something like mental health resources and coding resources, that sort of thing. We also have the Mentorship Committee. So in this committee, they pair incoming students with upper year graduate students, so either master's or PhD. It's a really great transition to grad school and just meeting someone that's I'm just going to share the screen. Oh, sorry.
That's okay. It's just because it was a new. Get back to Zoom. Sorry. Where is Zoom?
Oh, I hit the meeting. I think you have to escape.
There. Oh, okay. Sorry.
Maybe you go new share at the top.
Okay, yes, that would work. Okay, and this one here. Sorry about that. Okay, let's get back forward.
Right, so the Mentorship Committee. Great to link incoming students with upper year students. It's also nice just to have someone that's gone through what you'll go through and just having a new connection. And then lastly, we have the I Wish I Knew Committee. So this committee holds a bunch of different panels throughout the year on a variety of graduate student and graduate life topics. So in the past, we've done a Biggest Mistakes panel and a Women in Science panel.
So now that we've told you a little bit about what Songs is, I just wanted to show you a couple pictures from some of our recent Songs events, just to give you a feel of like what these events might look like. So one thing that was really fun that we did in March 2023, and I think we're planning to do again this year, is tubing at Bowler Mountain. So with the generous support from the Neuroscience program, we were able to subsidize the cost of tubing for neurograds, and we all went out and had a really good time. We also recently did a hike at Kamoka, so that was in October, this month, 2023. So lots of people and their dogs came out.
Okay, and I'll just talk a little bit about the mentorship committee. So again, really great opportunity for mentors and mentees to connect. This past September, we had, or I guess October, we had a backyard hangouts. We do that every year where mentees can meet their mentors. I personally have loved this committee. I had a mentor my first year who was really helpful for me, and I'm currently mentoring 2 mentees. So I'm hoping to give them some good feedback and tips for their year. Also, not just non-academic, but we also have the academic information. So like we mentioned on the information sessions committee, they hold a variety of different sessions. So for example, we've done like thesis defense information, scholarship information, that sort of thing.
Another project that Songs has been working on is a podcast called Brain Matter Chatter. We created this podcast because we realized that there is kind of a high prevalence of mental health issues in academia, but that they're not necessarily always talked about. And so we wanted to create a safe space for people to discuss these issues. Myself and several other students who were either previously on Songs or, oh yeah, I guess they're all gone now, but they were previously on Songs and previously in the graduate program. I created this podcast and we interview students, faculty, and other experts to talk about things like Zoom fatigue, eating disorders, exercise and mental health, dealing with problems with your supervisors and things like that. So if you are interested in any of these topics, they're available on like Spotify, Google Podcasts, and all of those sorts of podcasting sites. Neuroscience Research Day, or NRD, is probably our favorite and biggest event of the year. It's a 1 to 2 day neuroscience conference that takes place here at Western University. We're really proud to be able to host a variety of keynote speakers who come in from other institutions. So last year we had Dr. Burke and Dr. Jocelyn from Sunnybrook and Sick Kids. We also have faculty speakers from Western telling us about their research. We have workshops such as career panels or science communication workshops. We have trainee presentations where you get the opportunity to win an award. We have a neuroscience art exhibit, a debate, and an EDI panel. And so we have the date set for NRD 2024 in February 22nd and 23rd, which is during the winter reading week. So if any of you are around, undergrads are more than welcome to attend. And it's usually a lot of fun.
Okay, so I'm just going to give credit to the people of Songs for this year. So this is the executive committee. So there's Haley, myself, Ava, Greg, Michaela, Cadence, Alice, and Anvita. And then these are our committee chairs. So we have Chelsea, Ray, Raphael, Nidhi, Pranshu, Dika, Leanne, and Kunj.
So that's all we really have to say about Songs. We hope this presentation was interesting. If you're interested in keeping up to date with any of the Songs activities, you can follow us on any of our social medias. We're usually at Songs UWO. One thing that is potentially helpful to some of you who might be applying for CGSM scholarships, as Brian mentioned, is we're going to be hosting drop-in sessions for students to come by and get advice from upper year PhD students or master's students who have gone through this application process before. So once we announce those dates, we can send that to Susan and Laura and they can pass that info on to you, and we're be happy to see you out there. And yeah, if you have any other questions about the program, Ash and I are happy to answer them. You can always email us at Songs or whatever. Yeah.
Thank you.
Yeah, thank you.
Right, yeah, sorry about the screen sharing for those that are online, if you can hear me. So next up, we have Jordan, a PhD student in the program, and he will tell you about the Pasternak Lab.
Close these other ones.
Oh.
Get that other
Should
one
the sharing
so
go?
the
Yeah.
right one is shared.
I just do the screen.
We're gonna share the full screen.
You guys are better at this than me. I don't do this.
Maybe if you could just check if it's on, it's actually.
Yeah, I'll
Let's
check.
just do full screen. Oh, wow.
Yes,
I
it's good
don't-
now.
I had those meeting controls. More, yeah.
Boom, all right.
There you go.
Well, hi, everyone. My name is Jordan. I am a fifth year, PhD candidate in the Pasternak Laboratory over at Robarts Research Institute. And today I kind of want to talk to you about my lab's work and also just a little bit broadly about wet lab work here at Western. So broadly, our lab is interested in investigating the role of the subcellular compartment known as the lysosome in different neurodegenerative diseases. We're specifically interested in this as lysosomes have been found to play a role in toxic protein aggregate production. So Alzheimer's disease thought to be very important in the production of amyloid beta and the aggregation of amyloid beta and tau, as well as in other diseases such as Lewy body dementia, where it's thought to be involved in alpha-synuclein aggregation. So another big thing observed in many of these neurodegenerative diseases is that there's found to be a failure of lysosomal clearance of these toxic aggregates, and that's thought to play an important role. So our lab is very interested in investigating the lysosome in regards to, number one, how aggregates that are produced in the lysosome are then spit out of the cell through exocytosis, where it can then be potentially taken up by other cells. We're also interested in how things get to the lysosome. So how a cell surface protein, such as amyloid precursor protein, or extracellular aggregates, such as amyloid beta and tau, get to the lysosome through endocytosis. Finally, we're also interested, once in the cell and in the lysosome, how exactly cells or aggregates are taken up by autophagy and then degraded within lysosomes. So to investigate a lot of these things, our lab tries to use cutting-edge microscopy to try to visually look at these processes in cells and investigate kind of their roles. So for our investigations into exocytosis, we use this form of microscopy called turf microscopy, which basically allows us to have really good resolution just at the plasma membrane of cells. And what we then investigate is when cells are loaded with amyloid beta or produce amyloid beta endogenously, how lysosomes spit out this amyloid beta into its extracellular space. Additionally, and when we look at the endocytosis to lysosomes, what I do specifically, we use laser scanning confocal microscopy to either, in one of my projects, look at how APP found at the surface of cells is internalized to lysosomes where it's then produced into amyloid beta, or how a human neuron may take up extracellular aggregates like amyloid beta and tau inside of its cell where it may then seed more and more aggregation. So this is kind of some of the basic science in vitro work that we kind of carry out in our lab, but we also do carry out a little bit of in vivo preclinical work as well. So my lab mate Adriana, she's currently running a project looking at utilizing this treatment to try and boost lysosomal function to reduce the amount of amyloid beta produced by an Alzheimer's disease mouse model. So what she's found in very new evidence that treatment with this drug has actually drastically reduced the amount of amyloid beta in the brains of these mice by specifically increasing the amount of this protein known as glucocerebrosidase A. So it's a very important lysosomal enzyme, seemingly plays an important role in the reduction of amyloid beta. So these are ongoing projects, but there are many planned projects both in the basic in vitro work and as well as in these kind of in vivo preclinical models. So just to kind of shift gears too, I wanted to kind of talk about more about grad school. So first to start out, why you might want to consider doing wet lab work for your master's or your PhD. So first, it might be well suited to you if you have a desire to investigate and manipulate the nervous system from the most fundamental small basic units. So if you're interested in proteins, genes, and cells, wet lab work is very well suited for you. Additionally, if you also enjoy developing and refining physical skills and enjoy manual work. So while other types of neuroscience might offer you, the opportunity to intellectually investigate different ideas, wet lab work has a particularly interesting role in that you actually also get to develop very physical and very physical skills that if you're kind of a hands-on learner, you may enjoy. And ultimately, if you're interested in observing what can't be seen by the eye, well, microscopy work like we do is not a bad option. So initially, I just kind of like to talk broadly about kind of what I personally have learned and gained through my now 5 years of graduate school. So the first is patience and the importance of taking your time to develop a practical approach. The big thing you'll learn quickly in grad school is there is such thing as doing too much work. A very well thought out planned experiment running it once and seeing if it works is far better than trying to do everything all at once and not knowing where you are. Ultimately, like Brian talked about earlier, a big kind of main skill you learn to develop is organization and management skills. Coming out of undergrad, I honestly fundamentally did not have these. You can't exactly cram the day before for your PhD. It doesn't exactly work like that. So for me, this is something I struggled with at the start, but I have significantly improved my abilities to do these things, and these things will really help me moving forward in whatever career I may pursue next. And finally, resilience. So the big thing about science is you are going to fail, and you're going to fail time and time again. It's science. You're on the cutting edge. The first time you try something, it's not going to work. And that's okay. That's expected. It's built into it. So learning about how to deal with failure and learning how to learn from your mistakes or a failed experiment is by far the most important thing. So finally, just to close out, our lab is recruiting. So our lab is headed up by Dr. Stephen Pasternak. He is a clinician scientist, both here and at Parkwood Institute. And currently our lab has actually grown a lot in the past year. It used to be me and Adriana, shown by our pictures while it's just us two. But the two of us are hoping to graduate in the next year. So work based on our projects and other new projects we're picking up in the lab, especially a lot of the work around alpha-synuclein, are looking for new graduate students to take on. So I've included Steve's email here as well as my email, Twitter, and the barcode there will take you to my LinkedIn. So please feel free, if you're interested at all, to reach out to me. If you want advice about grad school and the application process in general, I'm always happy to help out and chat.
Great. All right. So, I think, Jessica, are you next, I believe? Yeah. All right, so next up, we have Dr. Jessica Grahn to tell us about some projects ongoing in her lab.
It should be already sharing.
Yeah, it's already shared.
Okay. Okay. There we go.
Will that unshare it?
It should.
It's, yeah.
Okay, good. All right, yes, my name is Jessica Grahn. I also did a neuroscience degree as an undergrad and a grad student. That's often not the case with neuroscience. People do come from all over, and that's one of the things that I really like about it. So my lab is interested in questions related to music and neuroscience. My core interests really are in rhythm and rhythm perception and the links between rhythm and movement. Why do we move to sounds in our environment when we don't move to most other sounds in our environment? And the neural mechanisms of that. And that whenever you have a single question, neuroscience is great because you have lots of different methods you can use to actually tackle those questions. However, I'm also someone who's driven by the interests of my students. So along the way, we've really expanded. We're looking a lot at sort of applications of what we know about how rhythm activates the movement system to gait. But I also have lines of research in relationships between music and language. What can learning about the structures of one tell us about the structures of other? Where's the blurry boundary between the two? What can the brain tell us about where that boundary is and how things are processed? Music and memory. So everyone has seen these amazing videos of people who are very far along the stages of dementia, and then they hear some music and suddenly they're talking and retrieving memories when seconds before they were catatonic. So what's going on there? And that's something that's very deeply tied into the brain. So we have lines of research in that. We also have people who are very interested in dance, which makes a ton of sense. I have no expertise in dance, but I do in the brain. So that's what people come, they bring the dance expertise. So I'll tell you a bit about the lines of research we're going on, but I am open to ideas that people have. The closer it's related to something I've kind of done before, the easier it is for me to supervise. And everyone has different levels of how much they're part of a core focus of the lab or more peripheral. I spent much of my life being peripheral to groups that I was part of because music is just not one of those things. There's many music and neuroscience centers. So I'm quite comfortable with that some people less so. It brings some benefits, which is you really own your work and you know what your project is all about, and you're not constantly looking to someone else to answer other questions. But that knowledge is hard won. So it takes a lot more going down rabbit holes and figuring things out for yourself and having to do a bit of your own networking to sort that out. The nice thing about graduate school is there's usually room for all sorts of people along all of these different dimensions. And the good thing too is that we grow. So we might not be very comfortable at the beginning, but by the end, as we've learned that we do have certain capacities and we're actually smarter than we think we are, that it becomes easier to branch out and do things more independently. So a lot of my research, as I said, is on the neural mechanisms of rhythm perception, and a lot of it derives from a very early study I did during my own graduate work, which was looking at people listening to rhythms and not moving, not tapping, not counting, and looking at the brain areas that responded and finding that, yes, okay, you get auditory cortex, surprise, surprise, but all the other areas that were lighting up, supplementary motor area, premotor cortex, basal ganglia in the core of the brain, cerebellum, are really areas involved in the selection, initiation, timing, and control of movement. So that drove a lot of future research. One thing about fMRI is it's correlational. So we've been looking at causal methods as well. One model of basal ganglia function is to look at people with Parkinson's disease. So we look at how their rhythm abilities are affected by the changes in their basal ganglia. But in healthy people, you can also use electrical stimulation, where you have a very low amperage current through a particular brain area to see if it changes performance. So unlike neuroimaging, which is still, although it's very sexy, very correlational, this is actually a causal method that we can use in healthy people, where we can actually change excitability or responsiveness in a brain area and see if it affects behavior. Lo and behold, it turns out if I ask you to try and detect changes in rhythms, again, not tap along to them or do any motor thing with them, just listen and detect if two are the same or different. When you're under sham stimulation, you do a certain level of ability there. But when I inhibit the supplementary motor area, one of those motor areas that lights up, we see you do worse. And when I excite it, we see that you do better. So this suggests that the motor areas are not just lighting up for the heck of it because maybe we're thinking about moving, but they're actually doing something core to the task. Now, what could that be? A lot of our models of timing, which is, I think, what's going on here. In order to perceive a rhythm, you have to be timing all of the individual time intervals that go into it, so much so that you can then see the relationships between them and work out if there's like an underlying beat structure that is actually structuring the entire rhythm. A lot of our timing research is based on non-human animals who do not really seem to show much evidence of beat perception. So is this moving to music thing something like a special adaptation of our timing system, is very species specific? Or is it something that exists in other species, but we just haven't got the right paradigms to observe it? Well, this is one of the few times when neuroscience can actually be useful, and looking at someone's brain instead of their behavior can tell you something you didn't already know, which is if you look at what happens in the brains of macaques, which is a study that we did in the pilot form about 10 years ago now here, do they show motor related responses to rhythm? And indeed they do. So the picture of a macaque brain with some beautifully outlined putamen. We're carrying on these studies now with marmosets. The downside of the macaques is they had to be anesthetized to be in the scanner, which meant that although we got beautiful results in the initial few minutes of the scan, they very quickly dropped off into noise because of the anesthesia. With marmosets, we can actually scan them awake and still, so we're going to be able to look at their responses. I've also done some work in the past with birds. If you're looking for a species that might show the ability to detect the underlying structures of rhythms, birds are a good bet. And we did some interesting work there that I'd be happy to tell you about sometime. And then the other thing that we do, as I talked about, is a bit on what are the applications of music to walking. So we can look at this in healthy people, exercise, does it give you more stamina? Does it help you pace yourself? All this stuff. But we can also look at how it affects movement with an idea toward applying to something like Parkinson's disease, where movement problems result. And for some patients, it seems to be very clear that a steady, regular rhythm really restores some of their normal movement. Why? I'm very interested in the neural mechanisms of this. Part of the issue in studying it is if you want to put people in a scanner, suddenly they're not walking around anymore. So we can use FNIRS, and this is a portable FNIRS cap, allows us to look at cortical brain activity. So we can't really look at the core structures deep in the brain yet, but we can look at cortical areas to work out what is driving this improvement for the patients who show improvement. One tangential thing I'll say is that one thing we're very interested in is people's individual differences in their ability to feel that beat and whether this predicts which patients are actually going to respond to having a regular beat help them. So just because we're at the halfway, a little more mark, and you need, all the help you can to stay awake, I'm sure I'm gonna give you a little task that we give our participants and patients to see how good their beat perception is. And here's some music and you just tell me if the beeps that go on the music are on or off the beat. Oh, we might not have sound. That often happens. Okay, well, come work in the lab and then you'll find out all about it. My lab website is there. My trainees are all listed on there. Feel free to contact them. They can tell you a lot about lab climate and culture. I work very hard to foster an inclusive climate. About half of our graduate students and postdocs are international. And we try to adhere to most of the best practices that we possibly can in terms of equity, diversity, inclusivity. And we're always looking for more people to come offer their perspectives. So thank you.
Great. All right. Good. Okay, so next up, we have Dr. Bruce Morton to tell us about his research.
Yeah, I've really enjoyed these presentations. Gosh, if I had a chance to do a PhD all over again, Warren, I just wouldn't know what to study. Anyway, yep, Shazam. Okay, so hi, my name is Dr. Bruce Morton. I'm a professor in the Department of Psychology. And I'm also a principal or core faculty member of the program in neuroscience. And it's really a pleasure for me to have a short opportunity here today to talk to you a little bit about the work that we do in my lab. I don't have anything fancy. I don't have dance along videos. I don't have these amazing, micrographs, and I don't have pictures of baseball games. But I do have this. Which is a short summary of one of the two studies I want to talk to you about today, and that I'm actively recruiting new students, hopefully one of you, to help out in. So for those of you who've witnessed changes in your in-laws' behavior, like maybe you saw that nephew or niece that it was so sweet and engaging when she was 8, and then you just saw her recently and she's 13 now, and she's like doesn't talk, and she's just on social media all the time and seems to be in a foul mood 80% of the time. And you're like, well, what's happening? What's happening as kids transition from childhood, the sweetness of childhood, into the complexity of adolescence? What's going on there? We see profound changes in emotion. We see profound changes in their assessment of risk, in their decision-making, and a whole variety of other things. And one of the things that we're doing in our lab is using MRI. So we're using one of these fancy machines to take these beautiful images of the brain. And we're using these images to study parts of the brain that are involved in dopaminergic signal transmission. So dopamine is a very important neurotransmitter that has been linked to a variety of these aspects of psychology that we're interested in: reward-based learning and decision-making, risk-taking, motivation, salient processing, and so on. Dopamine seems to be intimately involved in all of these things. And so we're hypothesizing that there are important changes in the dopamine system that have their onset in early adolescence. And we are very interested and motivated by the prospect of using MRI to take these images of the brain and analyze these images and gain some insight into the nature of changes that are taking place in kids as they transition into adolescence and from adolescence into early adulthood. Another related topic, again, broadly speaking, my lab is interested in what we might describe as self-regulatory processes. So how do you make decisions? How do you regulate your emotions? How do you formulate plans? How do you switch what you're doing when what you're doing isn't effective and you wanted to do something different? How do you introduce that kind of flexibility into the way you're going about doing things? All these kinds of functions that might loosely fall under the banner of self-regulation are things that we study in the lab. So one very everyday example of self-regulation is the regulation of our motivation to ingest really, really highly palatable, but not so healthy foods. Okay, and we are constantly bombarded with images of these foods. So if you're at home watching a sports game, probably you're going to see an advertisement from a local fast food outlet, and indeed you could press your whatever. What's the name? Uber Eats, for instance, yeah, or skip the dishes, and you could have that food item delivered to your house in 15 or 20 minutes, right? So it's these are just images, but they can have a profound impact on our behavior and on our craving for that food type. So we're interested in trying to understand, well, what cognitive and neurophysiological mechanisms do we have at our disposal to regulate the impact of food imagery on our cravings? And so what we're going to do is we're going to do something like Jessica Grahn, because every good lab at Western imitates Jessica Grahn. We're going to temporarily silence an area of the brain on the lateral side of the prefrontal cortex that's known to be involved in the regulation of thoughts and behaviors. We're going to temporarily silence that area using transcranial magnetic stimulation. And then we're going to put the participants in an MRI scanner and expose them to all these very beautiful pictures of foods and look to see how does the brain formulate a representation of these food images when these regulatory areas of the brain have been silenced, and compare that to the patterns of activity that we see elicited by these images in the brain when this regulatory area has not been silenced, right? So to get some understanding about how food images impact reward and regulatory areas of the brain, how these regulatory areas can modulate the representation of these food images, and hopefully learn something more about the cognitive neuroscience of dietary perception or food perception and dietary decision making. So yeah, so if you're interested, you would just want to talk about the studies, or you're perhaps interested in graduate opportunities in the lab, you're more than welcome to contact my lab coordinator or me. I'm going to have to bolt pretty soon, but hope to hear from you.
Perfect,
Great, thanks.
Thank you. Lost on me that there's pizza over there right now. Seems suitable. Okay, so that's it. Basically, we have some time for questions and answers. I don't know if there's any online as well. Yeah, are there any?
There is one question online. How
Fire
long is
away.
the graduate program?
Pardon me?
How long is the graduate program?
So how long is the graduate? It's 2 years for a master's, 4 year for a PhD. And if you could do a rollover, which is rather than 2 + 4 for 6, you could do 5 years for a PhD without a master's. Okay, let me just end that.
It was.
Don't want to do that? Yeah. Yeah, you guys are like watching me. It's like watching old parents do something I know on technology. It's terrible. Yep.
The other is a comment. It was, you mentioned it as course based, but that's not correct.
Yeah, sorry, there's not a course-based master's in the neuroscience program, but as part of your degree, you do take a couple of courses. Some are mandatory that everybody takes, and then some other courses you take depending on your specific area of research. Okay, yep.
So if you were to apply for the CGSM, was
Yep.
it? What's the timeline look like for that? Like, do you have to have a supervisor like fully secured before?
Yeah, that's a good question. I'm not sure actually, and I think that'll be something. Do you know,
I
Jordan?
roughly know. So if you're currently doing a thesis project, as far as what I'm aware of is you can write a CGSM with them based on a proposal and a project that they have. If you win it, but you wind up with a different supervisor, it should still stay with you. So they don't necessarily give you the award to do that research. Writing the proposal is more of a demonstration of your abilities to write a scientific proposal.
So even
and-
if you apply with like a different supervisor to begin with,
I-
and then transfer over?
I believe even if you went to a different school, it should stick with you.
Okay.
As far as I'm now school allocations might come into play there.
I don't think you can change schools. I think you can change schools, supervisors within a school. Does that sound right?
Yes, that sounds right.
Okay.
That does sound
Good.
right.
Yeah, so just for those that were online, a question was about the CGSM application. You can apply for it with, let's say, a description of work you're doing right now. And if you get it, you can switch supervisors within the school. Okay. Yep.
Between like like the awards and also ways.
Yeah, so how does the questions about how does funding work within the program? So since we are such a large program, we have supervisors in so many different departments. The funding plan for a particular lab depends on where that supervisor's home department is. So if it's a supervisor, let's say within the Department of Psychology, the neuroscience funding plan is the psychology funding plan for that student. That way there's no within lab discrepancies, but the psychology funding plan is distinct from the computer science funding plan. So there's going to be some program discrepancies. All that information's on the website. And that's a very common question that I think next year I'll put in the facts because, yeah. Sure.
Sorry, I have a question for the students. I guess like when you were deciding on like where to go and kind of who to work with, what kind of factors were you considering and what became like the biggest part of your decisions?
What I should say your biggest one is talk to the students in the labs you're interested in. Get a feel for the culture, get a feel for what the environment is like outside of just the supervisor and the work. Those are the people you're going to be interacting with the most, the people you directly work with, by far the most important thing. So for me, I stayed in the lab I did my thesis project in. I already knew the person who was, at that time, it was just me and my lab mate, Adriana. So I knew I worked very well with her and I enjoyed Steve. I enjoyed the work. So for me, it was an easy choice in that sense. I would say by far the most important thing you need to do is talk to students in that lab.
Haley or Ash, anything to add? There's also Omar at the back there. He's the student representative. So he's another student from the program here. So
I would also say like to consider kind of the techniques that you can come away with in that lab. So for me, I wasn't necessarily interested in staying in research forever. So I wanted to make sure that whatever techniques I'm learning could be applicable to like an industry position. So like I prioritized a lab that's more computational personally.
And on that, I chose wet lab and cellular molecular neuroscience because I didn't know if I wanted to stay in the brain forever. I can always move different systems, look at different proteins and different types of cells. So that was a big thing for me.
For me, I'll just add on. So I stayed in the same lab as my undergraduate thesis. I do animal work with mice, so it was a good chance for me to see like, do I even like working with mice? And I do. Another thing I'll mention is my lab is very hands-off. There's like hands-on versus hands-off supervisors. Be sure to know like within yourself if you like to have more day-to-day supervision versus you're okay being more independent on your own. For sure, like adding to their points, talk to grad students in the lab to know what the environment is sort of like. So
If they're a clinician, they're going to be hands-off.
Bruce and Omar, anything to add? Bruce, go
I
ahead.
was just gonna say, I mean, one of the things that often governs students as they're thinking about ways to go is like, you know, they have a question and they want to go and sort of find a lab that complements their question. And although I think that's important, I think it's probably more important to go to an environment where you're going to learn something. So even if you're like, you know, I really like this guy and the students, the professor's really cool and all the students in it, but they're doing the stuff on lysosomes, but if you go there and you're like, they're really cool and I think I could really learn from these folks, that's probably somewhere you should be thinking of going, not like, oh my God, I wanted to study the brain or whatever, right? So.
I think something that I didn't consider when I was looking at my lab is like the stage, I guess, of the PI. Like my supervisor is very young, like this is his first lab. And so I think there's like kind of differences if you're coming into a really established like lab where they might have like certain protocols already set up, whereas like a newer lab, like you're going to be doing a lot of that, like building stuff, pipelines and all that. So
That goes for projects as well.
Yeah,
You pick up a
projects too.
project a former student worked on, it can be, you know, a bit smoother sailing, but you know, you don't learn the things you do troubleshooting something from the ground up. So it's all what you want at the end of the day.
Omar, anything to add?
I mean, basically not everything that we've talked about here, these are all things you can and should ask the students because they're going to be very forthcoming and they're not going to lie to you about it. We're all in the same boat here. There's like a sort of camaraderie between us and, you know, we're not gonna lead you astray and put you in a position if we can help it that we don't think that you will thrive in. So, yeah.
Any lab that says don't talk to my students, that's a red flag. So I don't think any labs say that. Yep.
So the PhD length of it being 4 years, doing a 5-year thing, is that strict across all labs you're supervising?
So that's the sort of what's called the funding window. So your funding package is made up of all sorts of streams. Your supervisor gives you a stipend. You may TA, that's a source of funding. Some money comes from the faculty as well. So those 5 years, let's say if you do a rollover from a master's to a PhD, those are the 5 years where you can TA and get that sort of faculty funding. Some projects just take longer and so you might have to stick around for a little bit beyond that. And so that's just the funding window. We don't like kick you out on 5 years plus a day. But it's, you know, it's in everybody's interest to finish somewhat close to on time.
The big thing with that would be WGRS.
Yeah, that's the
window for
faculty,
that.
yeah.
So the money you get from the school that basically just goes towards your tuition, so you don't have to pay tuition, is the thing that is limited by that funding window. Which of course, if you have to go over because your project's going over, that's when you have a discussion with your supervisor, and often they will cover those fees for you to ensure that you're not missing out on that.
Sorry, is it possible to do it in a shorter amount of time?
Absolutely. Yeah, definitely. Omar?
I will just say that for a lot of people who are in the program now, they sort of have like an inside out approach where they already had connections like during their undergrad and sort of knew who they would want to work with or wanted to. I didn't have that. And so I did like the outside in thing where we actually have, I think, still a page with a list of all of our neuroscience faculty, links to their website. They have like selected publications you can read and sort of make a short list of who would be interested in working. So that's what I did, and it's a good way to start, figure out who you might want to talk to.
Yeah, there's a question from online about whether you can decide to do the rollover after you enter a master's. And in fact, that's the usual way it goes. So we sort of view that kind of first year as a master's. It's a bit of a probationary period, but I want to emphasize that probationary period works two ways. So that's the supervisor making sure you're a good fit for the lab and you making sure you're a good fit, like that the supervisor is a good fit for you. You know, there's no signing in blood. This is not like you only enter those things if there's two people, or sorry, yeah, both sides want that to happen.
So I guess what are sort of the, so obviously, I guess like talking to different supervisors to understand like where you want to work with next year is like a okay thing to do, but what are sort of like the, I guess like what's the etiquette about like I guess making it so like you don't sort of commit to one thing without like knowing where you're going to be ending up, I guess, if that's an okay question.
Like, I think you're welcome to probably interview with as many interested supervisors as you can. Just don't tell them you're going there until you know for sure. I think that's it. Like you can just be open that I'm just interviewing around, and you can let them know that I still have this many interviews. I'm expecting to make a decision by this date. And if they were in desperate need of signing someone up now, then they can tell you that and you work around that. But usually they shouldn't be too crazy about.
No, definitely not. It's in everybody's best interest to have a good fit. Definitely. I would say as well with one thing that's unique about the neuroscience program, we don't really have a strict application deadline. The application opens on November 1st, but then it's a rolling admission. Like it basically goes almost into the summer. And that's because since we're affiliated with so many different programs, all programs have independent and different deadlines for when they want their applications in. So we're quite lenient and flexible that way. Yep.
Really quick, are these slides posted somewhere, or your emails where I can find all of you?
Good point. We have everybody's contact info.
Everybody's contact info, I believe. This is being recorded, so we can send it out to you if you want to have a copy of it.
So
We can post it somewhere where you can access it.
and when in doubt, just reach out to either me or the neuroscience office as well, and we'll put you in contact with everybody. Okay. So just neuroscience, Western University will take you to the that information.
Our email address is neuroscience@uwo.ca.
Is there a list where you guys have like the profs and their research areas that?
It's all on the website.
Oh, okay.
Why don't I know there was another question. I'll just jump to it right now. There's the grad. So I know there was another question.
Just kind of like focusing more on like the methods of like, you know,
Well, yeah,
physiology.
if you were like, say for me, I could have done mine in physiology and pharmacology. At the end of the day, it kind of becomes a name on a piece of paper. And of course, the courses and other things you have to do. So me doing the physiology pharmacology program, if I did that instead of neuro, I wouldn't do weekly neuro seminars. I wouldn't take the few required neuroscience classes. So I still get a breadth of learning across neuroscience, but my specialization doesn't change regardless of what name is on it. So the minute details are kind of in courses you take, those kinds of things.
So just online here, this is the themes that I was mentioning in my talks, and each one of these you can kind of, you know, drill down and find all sorts of different categories. So yeah, there's a whole bunch of us right there. And each one of the faculty members may be listed in multiple locations as well. So we also have it listed alphabetically, but there's 100 of us, so that's a little bit harder to find.
On your question, I just thought, also, what you decide to do, if you decide to do neuro, physio, pharma, psych, it doesn't necessarily determine what you do next. My father's cardiologist has a master's in neuroscience. As far as I'm aware, there's no brain in the heart. So it's not necessarily, again, like think about what type of thing you might want to do, maybe more so than what's on the paper, and consider courses and what else is involved in the program.
Yep.
Is there a day where everyone gets their acceptance or reject, or do we just know as we go?
Yeah, it's already, you know, as you go, basically. It's a rolling assessment, basically. And it's like I mentioned before, it's a two-stage process. So the first thing you do is you apply for the program, to the program, and the program committee meets and we assess applications to see if it's academically acceptable. Then the second stage is finding that fit within a lab. So that's really where the bottleneck is. We have many more students that are found to be academically acceptable than actually place in a lab. And that's because the lab placements are determined by, you know, the particularities in that lab, funding, resources, projects, and things like that. Yeah. Oh, that's a good question. So we do these types of open houses, and it's I should say it's the application that opened on November 1st. So you can go start talking to people right now, and it's essentially the portal for applications opens on November 1st. I don't know if that's answering your question. Sorry, do we have other events? I think periodically there's sort of open house. I mean, this is one of them for sure. What other things would you think of? Yeah.
Like there's.
Yeah, that's true. There's an open house in February. That's right. Yeah, thank you. It's actually affiliated with the Neuroscience Research Day. So it's during reading week in February, and that's another opportunity to kind of come in. But you know, I think again, I just emphasize there's nothing preventing you from reaching out right now. And if the lab's interested, they'll arrange a visit with you so you can tour the lab.
That is by far the main thing about figuring out what lab you want to be, is just emailing supervisors, having an interview with them. They'll take you around their lab, show you around, offer to, whenever we have a new student come and interview with Dr. Pasternak, he brings them to us and we chat with them for a little bit too, show them around the lab. So, and the big thing is if you email a supervisor and you don't get a no back from them, I know I'm not taking students. If you've just heard nothing, that is not a no, keep emailing. It took me a good 3 emails to get a reply from Dr. Pasternak. And again, it can just come down to them just your email showing up in their cloud of unread emails. So until you get a no, keep reaching out if it's someone you're interested in. They will get back to you.
It's not the truth.
It's just the reality.
I know that. Thinking back to all my unanswered emails. Okay, so we're happy to, I'll stick around for a bit. I don't know if any of the graduate students can stick around. Happy to have sort of informal one-on-one conversations. But please, I'll tempt you with the pizza once again, help yourselves. And thank you so much for coming. Appreciate it.